Welcome to the learning module for DNA mutations and DNA repair. This is Dr. Tefke for the Cellular and Molecular Medicines course. After completing this learning module, students should be able to distinguish the different kinds of DNA mutations from one another, describe the process of direct reversal, explain base excision repair and give an example, describe nucleotide excision repair and give an example, explain methyl-directed mismatch repair, explain how defects in DNA repair can lead to cancer and provide specific examples, explain how DNA strand breaks are repaired, describe chromosomal rearrangements, explain how transposons contribute to genetic rearrangement and possibly to disease. Any permanent change in the DNA sequence of an organism is a mutation. Mutations are the sources of altered versions of genes and provide raw material for evolution. Most mutations do not affect the phenotype of an organism because large portions of the genome is non-coding DNA. Only a small percentage of mutations actually cause a change in our phenotypes. The simplest types of mutations are substitutions or base changes, where one base is converted to another. These can be classified as either transitions, where one purine is changed to another purine, an adenine to a guanine, for example, or one pyrimidine is changed to another pyrimidine, thymidine for cytosine, for example. Or transversions, where a purine is substituted for a pyrimidine, or a pyrimidine is substituted for a purine, for example, adenine to cytosine. Another simple type of mutation is the gain or loss of one or a few bases of DNA. These are referred to as insertions and deletions, and if they land in the coding region of a gene, can result in a frame shift mutation. Larger mutations can include insertions of whole new sequences, often due to the movements of transposable elements in DNA, or to chromosomal rearrangements such as inversions or translocations. Deletions of large segments of DNA can also occur. Mutations can also be classified according to their effects on the protein or messenger RNA produced by the gene that is mutated. Some point mutations can affect the DNA sequence but not the protein. These are called silent mutations or synonymous mutations. They are due to the fact that the genetic code is degenerate and several codons specify the same amino acid. Silent mutations affect the DNA sequence but not the protein and therefore have no effect on the organism's phenotype. Missense mutations substitute one amino acid for another. Some missense mutations have very large effects, while others have minimal or no effect. It depends upon where the mutation occurs in the protein structure and how big a change in the type of amino acid that is specified. Missense mutations are also known as non-synonymous mutations. Nonsense mutations convert an amino acid codon into a stop codon. The effect is to shorten the resultant protein. Sometimes this has very little effect. However, oftentimes nonsense mutations result in completely non-functional proteins. Sense mutations are the opposite of nonsense mutations. A stop codon is converted into an amino acid codon. Since DNA outside a protein coding region contains an average of 3 stop codons per 64 codons, the translation process usually stops after producing only a slightly longer protein. Insertion or deletion of a base within a coding sequence results in a frame shift mutation. Frame shift mutations result in all amino acids downstream from the mutation site being completely different from the wild-type protein. These proteins are generally non-functional. Therefore, frame shift mutations are much more deleterious than point mutations. The sources of DNA damage can be either spontaneous, arising from endogenous events such as deaminations, which removes an amino group from a base, depurination, which is a spontaneous hydrolysis of the glycosidic bond resulting in an apurinic site or AP site, or spontaneous oxidations, which add an oxygen atom to a base. Deaminations, depurinations, and oxidations occur quite frequently within cells, and if they were not repaired, further DNA replication would introduce transitions and transversions into daughter cells, and the changes would become permanent. Other mutations are the result of environmental or physical factors, such as alkylating agents, X-rays that induce single and double-strand breaks in DNA, dietary carcinogens, exposure to ultraviolet light, or carcinogens in cigarette smoke. Deaminations are potentially deleterious events since they change the base pairing properties of the affected base. For example, spontaneous deamination of adenine yields hypoxanthine. Hypoxanthine base pairs with cytosine, so further replication will convert the original AT base pairing to a GC base pairing in daughter cells. Spontaneous deamination of guanine will yield xanthine, potentially harmful because xanthine does not form stable base pairs with either cytosine or thymine, so DNA polymerase essentially has to guess which nucleotide to insert at this location when replicating DNA. Deamination of cytosine yields uracil. Cytosine pairs with guanine, but uracil pairs with adenine. So continued replication can produce a daughter cell with an AT base pairing at this location instead of the original CG base pair. There is no need to memorize these potential changes in base pairings. These examples are only provided to illustrate how changes in base structure can lead to permanent mutations. Depurinations can occur from spontaneous hydrolysis of the glycosidic bond between the deoxyribose and the nitrogenous bases. This will leave an abasic or apurinic site on the DNA strand. When DNA polymerase uses this strand as template, it does not know which nucleotide belongs at this position, so it randomly inserts one and continues on its way. Reactive oxygen species can also modify DNA bases. For example, guanine can be oxidated at the 8 position to generate 8-oxoguanine. 8-oxoguanine base pairs with adenine instead of cytosine. Exposure to ultraviolet light can induce adjacent thymine bases on a DNA strand to become covalently bonded to one another. This is referred to as formation of a thymine dimer. During replication, the dimer can be replicated as a single base, resulting in a deletion. DNA is the only macromolecule in our cells that is repaired. Everything else is simply replaced. The human genome has over 100 genes dedicated for DNA repair, and even small organisms have numerous DNA repair genes. Cancerous transformation of cells is a consequence of inadequate DNA repair. Direct repair mechanisms, or damage-specific repair, is typically utilized to remove alkyl groups on DNA. For example, the O6 position of guanine can become methylated, which would allow it to base pair with thymine instead of cytosine. The enzyme O6-alkyl guanine transferase, or AGT, directly reverses this alkylation of guanine to repair this DNA damage. As the reaction inactivates AGT, it is a suicide enzyme. Each AGT enzyme is only able to catalyze a single reaction and then is destroyed afterwards. This is an expensive process for the cell. However, the prospects of keeping the damaged DNA in the genome outweigh the high metabolic cost of synthesizing the AGT proteins to reverse the alkylation events.
chemical agents can induce the methylation or alkylation of guanine residues in DNA at the oxygen attached to the 6th position of the purine ring. These methylation changes can result in mutations. The enzyme methylguanine DNA methyltransferase eliminates these methylation events and restores the correct structure to guanine in DNA, preventing mutations from occurring.
The most common means of repairing damage or a mismatch is to cut out the duplex DNA and recopy the remaining complementary strand of DNA. Two different types of excision repair have been characterized: base excision repair and nucleotide excision repair. Base excision repair is utilized for correction of small damaged bases which do not significantly distort the DNA double helix. Nucleotide excision repair is used to remove larger damage that results in a distorted DNA helix. They both utilize what is referred to as a cut, copy, and paste mechanism, which has 4 steps. First, the damage has to be recognized. Then an enzyme or complex removes a damaged base or a string of nucleotides from the DNA. A DNA polymerase will then copy the template strand to replace the excised or damaged portion of the opposite strand. The DNA polymerase can initiate synthesis from the 3' hydroxyl at the single-strand nick or gap in the DNA remaining at the site of damage after excision. Finally, DNA ligase seals the remaining nick to give an intact, repaired DNA. Some examples of the DNA damage repaired by the base excision repair mechanism include apurinic sites, methylated bases, and oxidized bases. A variety of DNA glycosylases are used to both detect and remove these altered bases. Cytosine can spontaneously deaminate to uracil. If this happens in RNA, not much of a problem because RNA can be made again. If it happens in DNA, it is potentially mutagenic. Base excision repair mechanisms utilize a uracil DNA glycosylase to convert uracil in DNA back into cytosine residues. Having thymine in DNA instead of uracil allows deaminated cytosine in DNA to be repaired by the base excision repair mechanism. The methyl group on thymine serves as a tag that allows the repair then of deaminated cytosine in DNA because thymine is not recognized by the uracil DNA glycosylase and removed. Base excision repair is initiated by DNA glycosylases, such as uracil DNA glycosylase, that recognize the chemically damaged base. The glycosylase then cleaves the glycosidic bond between the base and sugar, leaving an abasic site. The AP endonuclease can then nick the phosphodiester bond 5' to the abasic site. When DNA polymerase binds the free primer end at the nick, its 5' to 3' exonuclease activity cuts a few nucleotides ahead of the missing base, and then its polymerization activity fills the gap. DNA ligase then performs the final nick sealing step.
By a system called base excision repair. Base excision repair works differently from nucleotide excision repair, which clips out a section of DNA containing a pyrimidine dimer or other defect to replace it with new DNA. Base excision repair acts on other kinds of defective DNA. For example, certain bases that are not supposed to be in DNA, such as uracil, are removed by base excision repair. If the base cytosine spontaneously deaminates, it forms uracil. Uracil can also be incorporated erroneously into a strand during DNA synthesis. If uracil forms in the place of cytosine, it would have a mutagenic effect. Whereas cytosine forms a base pair with guanine, uracil forms a base pair with adenine. During DNA synthesis, DNA polymerase would incorporate an incorrect nucleotide into the growing strand. A few other types of damage are also repaired by the base excision repair system. For example, When adenine becomes deaminated, it forms an unnatural base called hypoxanthine. A mutagen can also damage adenine by alkylating it, converting adenine to 3-methyladenine. Normally, adenine forms a base pair with thymine, but hypoxanthine in its place is mutagenic because it forms a base pair with cytosine. 3-methyladenine is even more damaging. When it is encountered in the replication fork, it stalls DNA synthesis. Consequently, repairing these mutations is vital for survival. Base excision repair employs a battery of glycosylase enzymes that can recognize and clip damaged bases from the phosphodiester backbone. Hypoxanthine DNA glycosylase, 3-methyladenine DNA glycosylase, and uracil DNA glycosylase recognize hypoxanthine and 3-methyladenine and uracil respectively, when these bases are present in DNA. The glycosylases cleave the bond connecting the damaged base to deoxyribose in the phosphodiester backbone. The result is an intact phosphodiester backbone missing a base. The site is called an AP site. In this case, it is missing a purine and is therefore an apurinic site. If it were missing a pyrimidine, it would be an apyrimidinic site. The next step in base excision repair involves AP endonucleases that specifically cleave the phosphodiester backbone at AP sites. The 3' end produced by the AP endonuclease provides a starting point for DNA polymerase I to start adding nucleotides. At the same time, the polymerase uses its 5' to 3' exonuclease activity to degrade the cleaved strand downstream of the AP site. DNA ligase then seals the remaining nick and the repair process is complete.
Direct repair and base excision repair mechanisms both use specific enzymes to detect and remove certain kinds of modified bases. Nucleotide excision repair is a system to remove damage in general that causes a distortion in the helical structure of the DNA. These modifications include thymidine or pyrimidine dimers, bulky adducts such as those created by the carcinogen benzopyrene, or cross-linking agents like cisplatin. The DNA of a human skin cell exposed to sunlight will accumulate thousands of thymine dimers per day. If these are not removed by nuclear excision repair, they are potentially mutagenic. The process of nucleotide excision repair is similar to base excision repair. The damage is detected by multiprotein complexes. A section of DNA surrounding the error is removed by the activity of endonucleases. The resulting gap is then filled in by DNA polymerases, and the last nick sealed by the activity of DNA ligase.
Commonly encounter ultraviolet light, which can cause neighboring pyrimidine bases, thymine or cytosine, to form dimers. Thymine dimers are the most common. Pyrimidine dimers can be repaired by several mechanisms, including a method called nucleotide excision repair. In this system, a 3-subunit exonuclease, made up of the proteins UVRA, B, and C, excises a patch of 12 to 13 nucleotides spanning the dimer. ATP hydrolysis provides energy for many steps throughout the process. The repair mechanism begins with the association of UVRA and UVRB and the hydrolysis of ATP. The UVR-AB complex binds to the DNA as UVRA makes the initial recognition of the DNA damage. UVRA bends the DNA near the damage. With more ATP hydrolysis, UVRA loads UVRB onto the damaged DNA, after which UVRA is ejected from the complex. Next, UVRB binds UVRC. UVRC nicks the damaged strand at 2 sites flanking the damaged bases. Another complex, called UVRD helicase, strips the resulting short, single-stranded damaged fragment from the DNA molecule. The single-stranded gap that remains has a 3' hydroxyl group that allows DNA polymerase to synthesize new DNA. After DNA polymerase is finished, DNA ligase seals the remaining nick in the DNA. Although this example showed the repair of a pyrimidine dimer, cells use nucleotide excision repair to fix a broad range of DNA lesions.
Mismatches that escape proofreading during DNA replication can be corrected by a methyl-directed mismatch repair mechanism. The adenine in the sequence GATC is methylated in DNA strands by an enzyme called maintenance methylase. For a short period after DNA synthesis, only the parent strand will contain these methyl residues on GAT sequences. If a mismatch is detected, a group of proteins known as mute proteins recognize the mismatch and identify which strand is likely to be the parent strand based upon its methylation pattern. A portion then of the daughter strand containing the mismatch base is then excised by these mute proteins. DNA polymerase then fills the gap and DNA ligase seals the nick.
DNA, it uses proofreading functions to prevent the misincorporation of bases. However, DNA polymerase does incorporate an incorrect base for approximately 1 in a million bases synthesized, resulting in a mismatch that distorts the structure of the double helix. Curiously, far fewer than 1 in 10 to the 6 mutations actually arise. The mutation rate in an E. coli cell is only 1 for every 10 to the 9 or 10 to the 10 bases replicated. The cell has a number of repair methods, including one called methyl mismatch repair to remove and replace a mismatched base. Methyl mismatch repair involves recognition of the methylation pattern in DNA bases. E. coli methylates its DNA at the palindromic sequence GATC using an enzyme called deoxyadenosine methylase, or DAM methylase. DAM methylase adds a methyl group to the adenine in both strands of the sequence. Adenine and thymine bases pair normally. The DAM methylase acts soon, but not immediately, after replication of a DNA sequence. Where DNA has methylation sequences, the parental strand will be methylated, but the new strand will still be unmethylated. The difference in methylation allows the cell to distinguish the correct sequence, presumably on the parental strand, from the incorrect sequence on the new strand. A series of proteins called mut-S, mut-L, and mut-H act together to repair the mismatch. Mut-S first binds to the mismatch and recruits mut-L and mut-H. Mut-L recognizes the methylated strand and brings it in a loop. Mut-H then cleaves the unmethylated strand containing the mutation near the GATC. A DNA helicase called UVRD then unwinds the cleaved strand, exposing it to a variety of exonucleases. The result is a gap that is filled in by DNA polymerase I. The DNA polymerase continues through the region that originally contained the mismatch, inserting a correct nucleotide this time. After a while, the DNA polymerase falls off the DNA, and the gap that remains is sealed by DNA ligase. The methyl-directed mismatch repair proteins and genes are called mute because strange
Ionizing radiation, such as gamma rays or X-rays, can induce single and double-strand breaks in DNA. Double-strand breaks can be repaired by two mechanisms: homologous recombination and non-homologous enjoining. Homologous recombination requires the presence of an identical or nearly identical sequence to be used as a template for repair of the break. The enzymatic machinery is almost identical to the machinery that's used during homologous recombination during chromosomal crossover in meiosis. This allows a damaged chromosome to be repaired using a homologous chromosome as a template. Non-homologous end-joining is referred to as non-homologous because the end breaks are directly ligated without the need for a homologous template, as occurs in homologous recombination. Non-homologous end joining typically utilizes short homologous DNA sequences to guide repair. These microhomologies are often present in single-strand overhangs on the ends of the double-strand breaks. When the overhangs are perfectly compatible, the repair is usually very accurate. Imprecise repair leads to the loss of nucleotides and can occur, but it is much more common when the overhangs are not compatible. Inappropriate end joining can lead to the translocations and telomere fusions that are hallmarks of tumor cells. There are several well-known diseases and disorders associated with defective DNA repair. All of these inherited disorders carry with them a predisposition for certain forms of cancers. For example, persons with xeroderma pigmentosum have inherited a defect in nucleotide excision repair machinery. Individuals with xeroderma pigmentosum have a predisposition for skin cancers due to the inability to repair UV-induced or sunlight-induced thymine dimer formation. Defects in mismatch repair enzymes are associated with hereditary non-polyposis colorectal cancer or Lynch syndrome, and inactivating mutations in the BRCA1 and BRCA2 locus leads to hereditary breast cancer due to failures in double-strand and single-strand DNA break repair mechanisms. Larger mutations are referred to as chromosomal rearrangements. Some common forms of chromosomal rearrangements include deletions, where portion of a chromosome is lost, duplications, in which portion of a chromosome is duplicated, and inversions, in which portion of a chromosome is excised and reinserted in the opposite orientation. Translocations occur when chromosome segments and the genes they contain change positions. Non-reciprocal translocations occur when the transfer is one way. Reciprocal translocations occur when two chromosomes swap parts. Transposable elements, or transposons, are sometimes called jumping genes because they promote their own movement from one location in the genome to another. Transposon activity can result in the formation of deletions and inversions and in the inactivation and activation of gene expression. There are two main classes of transposons: DNA transposons, which encode a protein transposase, which is required for their movement in the genome, and retrotransposons, which move through the action of RNA intermediates. Most transposons appear to be silent, not having an effect on the phenotype of the cell. However, there have been instances of a clinical outcome resulting from transposable elements. For example, insertion of a transposon known as L1 into the factor VIII gene has been noted to cause hemophilia. Here are a few multiple choice and true-false questions to test your mastery of DNA mutation and repair.